well. I am going to look at something very simple. So scale is the critical piece. Dorian laid out the distinction between symbiosis and symbiogenesis. And I'm simply going to talk about the scales at which symbiogenesis and symbiosis occur, and they're literally across the entire biosphere. So if maybe you can get me screen sharing, is that possible, Igor?
Up to you. You can definitely screen share.
Oh, I can do it. Let me see. I have to press a button probably. Let me see. There it is. Yeah, thank you. All right, let's take a look at a very few slides here. Oh, good. There it is. Okay. So just so we understand the scale, imagine, well, basically every discoverer of symbiogenesis would have looked at essentially this thing here. There's a chloroplast, which is relatively easy to see. And as you can see over on this other side, they're green. But then the hard part is actually to see also the mitochondria, which is essentially membrane bound inside. So those are on the order of a few to several microns. So mitochondria are generally smaller, and there's up to many thousands in a liver cell, and sometimes relatively few in a plant cell, and something like 100 plus chloroplasts in a plant cell. So the point is that the insight was to recognize that these structures at two different scales, basically from the larger cell that they inhabited, were different. And this was seen by many. What Lynn Margulis did was basically recognize that there was a genetic identity that demonstrably showed they were not of the same origin as the eukaryotic host. So that insight is basically what made possible the biology we live in now. And that invasion of this creature and these creatures, 1.5 billion years ago, literally changed the biosphere forever. So I worked on some of this. So that's symbiogenesis. I cultured many liverworts. So liverworts, they're small creatures, and you know, of course, the mosses, and the mosses are the much younger relative. Creatures like the liverwort are probably 400 million years old. And so what you're looking at here is something like 1 centimeter in length. So these things are the better part of 1 centimeter. These are the spores, basically, that be let drift into the air. But I want you to look closer here. You see these little dark spots. Those are cyanobacteria, nostoc. And it's extremely, so the problem with symbiogenesis is you don't want to bet on the wrong horse. So in America, the horse probably evolved, but then went extinct. And so you need something that's going to be essentially as eternal as you are. So every time you say the word vitamin, we indicate that that's a connectivity to an organism that does something that we cannot. And so vitamin C, we don't make it because the gorillas don't make it. The gorillas eat something like 4 grams of vitamin C every day in their diet, and we may have originally as well. So the point is that basically if your environment connects you with essential nutrients, then you don't need to incorporate it. But there's no way to do that, of course, for the bobtailed squid. And this creature, this is Blasia, has incorporated nostoc because in wetland environments, nitrogen is in low amounts. Therefore, you want it literally made for you all the time because you run out of it. And every creature you look at, every plant is about 15% of it is nitrogen. So a critical, absolutely critical building block. So symbiogenesis, symbiosis, it all occurs in here. So what Dorian was describing, so in the middle, in the beginning, there was only carbon dioxide as an electron acceptor. And methanogenesis was basically the way the world worked. So at some point in time, probably in the black smokers, you got sulfate reduction to sulfide that would bind metals, and that's another electron acceptor. And then the one that's critical when you get to exactly that environment you were looking at a second ago, where Blasio lived, where early land plants lived, you need nitrogen. And there's plenty of nitrogen gas in the atmosphere, N2, but basically you need a way to get that fixed. So that requires a cycle. I don't have the equation in here, but it's basically N2 and NO3 and ammonia are tied together by a couple of several bacterial groups. Oxygen kills all of these. So that was the event that Dorian was describing. Basically, when oxygen was invented, it literally put into hiding all of these creatures. They could no longer live in the open air anymore because the open air was now toxic. So the critical thing is there's basically, this is EH is an analog, they're akin to pH. So basically this is the electron availability, density. This is the proton pH3, H3O availability. Some very acid, so stagnant bogs in our stomach, to very basic, essentially peroxides and calcium carbonate and all. In this range is every reaction that you can name. And basically these are the drivers of evolution, because you cannot be an ungulate, you cannot be a mammal and have only fermentation going on, only nitrate. The waterfall is not well shown here. Basically a minus, and this is the extra electrons of 20.22 volts. Here it's basically on the other side, it says plus. In other words, there is a complete paucity of electrons, and you're running at a much, much higher level of electron volts. In here is all of life. And so something like our fermentation and lactic acid is someplace in here, and then here you get where there's nothing. So if you want to actually break down, literally sequester all metals, if you run it through one, doesn't matter what metal, lead, cadmium, mercury, you run it through a pile of limestone with a carbon source, all of the metals will be bound to the product of this reaction, which is hydrogen sulfide. Sulfate goes into sulfide, sulfide binds all the metals. That's where you get them now. If you want to mine gold, you get literally gold sulfide or sulfur sulfide out of the ground. And this is the range in which all organisms, all of life is ordered. And in some cases, and the one I just showed you, for example, you have such limits on nitrogen that you have to couple through symbiogenesis, literally the nitrate fixers with the carbon capturers. And there you have a symbiosis that's become a symbiogenesis. Often enough, you can have free living sulfate reducing and nitrate reducing bacteria, and they're in other kinds of structures that look kind of like this. So again, oxygen will kill all of the sulfate or nitrate reducers and all the sulfate reducers and all the methanogens. But basically the organisms on top, they can literally use something like zinc and iron as electron acceptors. And so you can have very low levels of iron, oxygen here, and lower and lower and lower till they're virtually zero when you get to reducing states like methanogenesis. So the point is that symbiogenesis and symbiosis occur across a huge range, but you can see that the very ordering structure which allows all of these organisms to live within something like this distance, centimeter to millimeters to centimeters, is essentially an interaction. And this is not actually symbiogenesis. The films develop everywhere. And you know this because we live off the microbial films that break sewage down in a relatively short order, and or anything else. Literally, there is not a single compound you can name, or I can name, where it's not mineralized or sequestered by the activities of microbial consortia. So another neat thing about these creatures, so they all, the land plants, have sometimes very close to symbiogenic. Actually, the Russian discoverer of the symbiogenic connection of mitochondria and cells studied lichens. And lichens are basically a weave. I should have brought a picture of them. They're right on my tree here. They're literally a weave of fungi and algal cells together where they're connected. They are, in a way, symbiogenic because the same species of lichen is literally always, most always, nothing is always anything in biology, most always actually the same species of algae and fungi. So those creatures are different because the fungi, excuse me, has much more surface area, and also can both protect the plant from fungal and bacterial invaders, but also can pick up water and nutrients, which is essentially the deal. Dorian didn't mention a great, but he implied basically the cooperative structure of nature, pointed out in a number of approaches. One great one was the evolution of cooperation by Axelrod from got to be in the maybe early 90s or 80s. And he showed basically through game theory arguments, you can always cheat, but mainly you get more out of the environment by exchange, cooperative exchange. So most everything is driven. So these creatures, I can tell you for certain, have a mucorrhalian mycorrhizal associate, as well as this nostoc. And literally every land plant you can see, if you take it and press it onto a petri dish with only methanol as a carbon source, you'll get a beautiful shape of the leaf, because the only thing that can live on that carbon source is a pink pigmented, this is hard to say, methylotropic bacterium that produces vitamin B12, required by every creature on planet Earth. So that's where I'll stop here. I want to say that there's basically across the scale of nature, from where we are to, let me try and see if I can get out of this. Thank you. From where we are at human scale to the scale of whole biomes, like the tropical rainforests, like the temperate landscapes of meadows and forests we live in here in up in the Northeast, like the Serengeti we'll see later. There's symbiosis between many of the collaborators there, but there's also symbiogenesis, especially in root structures, in soils. And we haven't looked, we only got the molecular tools to look as carefully as Dorian's description was laying out before, relatively recent history. So it's only with PCR, with being able to literally identify the genomic components from multiple ancestries, that we can actually get at this question. So soils are probably the best example of tightly woven symbiosis plus symbiogenesis. I don't think we've sorted that much out of it as yet. But again, every time, literally every time you say vitamin, every time we use that concept, it means that there is an organism someplace in our surroundings that basically creates something that has to connect with us, because our biochemistry requires riboflavin, thiamine, B12, vitamin C, the rest of them. So we don't want to recognize that basically it's not that we simply use this stuff. It's literally our life history is so tied with the organisms that produce these complementary compounds that we can't live without them. And that's true of a great deal of nature, that we would be best suited by literally entertaining in our surroundings.